Carrier proteins are pretty specific about which substrates are allowed to bind to them. So a glucose transporter allows glucose, and actually it also allows other 6-carbon sugars to bind to it, such as galactose. So galactose is another type of sugar that the glucose transporter will allow for it to bind to it. But there are some sugars like maltose. Maltose, it's a disaccharide. If maltose binds to that glucose transporter, it's not going to be allowed to get transported. It's actually going to get stuck. And so we refer to maltose as a competitive inhibitor because it's going to inhibit the transport of the molecules. It just blocked that carrier protein. So as I said, the glucose transporter can allow both glucose and galactose to pass through. Now the thing is, if only glucose was the only sugar found, notice how a lot of glucose is going to be transported. But if you have glucose and galactose around, then you're going to have a bit of competition between the two because glucose and galactose are both going to get transported through the same transporter. And so they're both going to be fighting to bind to that carrier protein. And so therefore, if you will take a look at the glucose transport rate, it's actually going to drop because now there's competition because you have galactose around. Now, carrier proteins are going to keep transporting those substrates either into or out of the cell. And so the more carrier proteins, the faster the transport is going to be for those molecules. But there will come a time where all of the carrier proteins are currently occupied with all of these substrates, and so the rate of transport is going to come to a plateau. And that's because all the carrier proteins are now saturated with the molecules that they are allowing their passage. Now, as we said, there is active transport and passive, and we said passive does not require any energy, but active transport does require energy. We talked about the primary active transport and the secondary active transport. Right now, we're going to talk about vesicular transport, which also is an active transport. So the vesicular transport that we are going to talk about, we have two types. One is known as endocytosis, endo meaning inside. So this type of transport is going to allow the transport of certain molecules to get transported into the cell. And we have two types. So we have a type known as phagocytosis and pinocytosis. Phagocytosis means cell eating, and pino means cell drinking. Phagocytosis and pinocytosis allows a huge bulk of molecules to pass through, whether those molecules are like solid particles, like food, for example, or whether the bulk is basically a large amount of fluid to be passed into the cell. Another type of vesicular transport that's also known as endocytosis is a receptor-mediated transport. So let's go ahead and take a look at a few examples. So this is the plasma membrane right here. And let's say that we have a bulk of particles that need to be transported, and those particles are pretty big to be allowed to pass through a channel protein or a carrier protein. Then how are these particles going to be allowed to pass into the cell? The plasma membrane dips inwards and it kind of folds around those particles, and part of the plasma membrane actually gets pinched off, creating that vesicle right here, which we refer to as a phagosome. And then If you want to break down these particles inside of the cell, there are usually organelles known as lysosomes that will go ahead and kind of merge with the phagosome, and they will allow the breaking down of those particles that just got sucked into the cell. There's also exocytosis. Exo meaning that the molecules are going to be pushed out of the cell. So if there are particles that want to exit the cell, but again, they're too big or you just have way too many of them, then passing them through a carrier or a channel protein is going to take forever. And so this vesicle merges itself, kind of fuses itself with the plasma membrane, and the plasma membrane opens up, allowing the exit of or the release of the contents. The receptor transport for these, let's say you have this substrate right here that wants to be transported into the cell, it will have to bind to a specific receptor protein on, let's say, the surface of the plasma membrane. Once it binds to it, what usually happens is that this receptor ligand complex will actually move along the plasma membrane until it locates an area within the plasma membrane that has some specific proteins known as clathrin proteins. Once it finds that area, so it will keep moving until it locates this region right here. The part of the plasma membrane right there is going to start to dip. And same thing, kind of same concept with the endocytosis. The plasma membrane dips, folds around the receptor ligand complex, and pushes it into the cell. Once it's inside of the cell, your cell can go ahead and dissociate or separate the receptor from the particles that your cell needs. And those receptors can actually get transported into a vesicle, and those receptors can actually get pushed back out via exocytosis so that they are going to be placed back on the plasma membrane. So those receptors can get recycled. The electrical state inside of our cell is more negative compared to the electrical state outside of our cell, and we refer to that as the membrane potential. The membrane potential inside of our cell at a resting state, which we refer to as a resting membrane potential, is around -70 millivolts. If we take a look at this graph right here, which is graphing the membrane potential inside of our cells, we notice that our resting membrane potential is around -70. If that resting membrane potential becomes more negative, we use the term hyperpolarization. So if it drops, let's say, to a -90 compared to a -70, we refer to that as hyperpolarization. If after it drops down to a -90 inside of your cell, it goes back to its normal state, we refer to that as repolarization, which means it's going back to its normal resting membrane potential. Now, if the resting membrane potential shifts from a -70 and now inside of your cell it becomes less negative, so the state shifts from a -70 to a -40, we refer to that as depolarization. Now, how does the cell maintains that resting membrane potential of a -70 millivolts? Let's say here is your cell right here. That purple membrane is representing the plasma membrane. Now, remember how we said we have different types of channels? We've got leaky channels, we've got gated channels, we've got carrier proteins. So this image right here is showing you that we have some leaky channels We refer to them as sodium leaky channels, and we also have potassium leaky channels. Those leaky channels are allowing the passage of sodium and potassium down their concentration gradient, meaning that sodium is going to move from the high sodium concentration outside of the cell into the cell. And potassium, since we have more potassium inside of the cell, potassium is going to be exiting. Okay, now if we take a look at the charges of sodium and potassium, they're both positive. Now, one thing to note is that your cell has more potassium leaky channels compared to the sodium. So if sodium comes in and it's carrying a positive charge, right? The more positive charge comes in, the more the cell could become positive on the inside. But to counter that, potassium is leaving, and potassium is positively charged. And since we have more potassium leaving compared to the sodium entering, the more positive charges leave, the more the cell becomes negative. But then how come if more potassium leaves, how come the cell does not become negative 90 or negative 100? Why is it maintaining that negative 70 and not becoming more negative? So to maintain that negative 70, the most important carrier protein that would help with that is the sodium potassium pump. With the sodium potassium pump, 3 sodium leave and 2 potassium go in. So it counteracts the exit of the potassium ions, because if a lot of potassium ions, as I said, leave the cell will become way too negative. But when you counteract that, pushing 2 potassium in and 3 sodium out, that maintains that negative 70 millivolt state. With this image right here, we're going to talk about how does your cell release insulin. So we know that insulin is produced in your pancreas. Inside of your pancreas, there are some cells known as beta cells, and that's where insulin is made and eventually released. Now, insulin is going to be released after you've eaten a meal. Or basically insulin is going to be released if the glucose levels in your blood is high. So if your glucose levels in your blood is low, insulin is not going to be released. And so what happens is once your glucose levels in your blood increases, that's going to force your insulin to be released out of those beta cells. How is that possible? So once your glucose levels in your blood increases, your glucose is going to be transported into the cell. And so that means that your energy level is going to start increasing, correct? After you eat, you feel more energized because the glucose is rushing into the cell via that glucose transporter. Now, when the ATP levels increase inside of your cell, ATP is actually going to bind to a potassium channel, closing that potassium channel. And when that potassium channel closes, what happens is the electrical state inside of your cell is going to differ. Remember how we said the resting membrane potential inside of your cell is a negative 70? And so when those potassium channels close, basically potassium is going to be prohibited from exiting the cell. And if potassium is prohibited from exiting the cell, what happens is the state inside of your cell depolarizes, meaning it starts to become less negative because now you have less positive potassium ions leaving the cell. Now, what happens is when your cell becomes less negative, that forces a voltage-gated channel known as the calcium voltage-gated channel to open, forcing calcium to enter into the cell. Once calcium enters into the cell, calcium is going to allow these vesicles to merge or fuse with the plasma membrane, and exocytosis of insulin will take place.